Swayam Prabha. Digital India. Educated India.
Hello everybody, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati, and in this module we were discussing about the characterization of the overexpressed products which you are getting from the host cells. And in this context, so far what we have discussed about the identification or the determination of the molecular weight. What we have discussed so far, we have discussed about how to determine the molecular weight of the isolated product. Either it could be by the gel filtration chromatography, which will give you the native molecular weight, or it could be by the electrophoresis, which will actually going to give you native as well as the subunit molecular weight. In addition, while we were discussing about the electrophoresis, we have also discussed about what are the different applications of electrophoresis, and in that context we have also discussed how the electrophoresis can be used in conjugation with the electroblotting techniques to determine or to check whether the isolated product has any kind of immunoreactivity with the antibodies or the developed antibodies. Now, the next thing what you can do in this series is you can deduce the sequence of the biomolecules, and that will be always been done by the sequencing techniques. That sequencing techniques will always going to tell you about the arrangement of the monomer, because in the case of biomolecule, whether it is the protein or the DNA, it is not important that you have all the functional group present, but it is also important that in what sequence these functional groups are being arranged. So, if you remember in the previous lectures, while we were taking up the conformation of the clones, we have discussed about how to sequence the DNA or the recombinant DNA what you are going to produce, so that you can be able to use that DNA sequencing techniques to verify the cloned DNA. Now, in today's lecture we are going to discuss about how you can sequence the proteins. So, whether it is protein or the DNA, the sequencing techniques for a biomolecule always follow flow chart, and when you talk about the biomolecules and the arrangement of the monomers, you have to have a schematic way of doing it, so that you will not be able to miss out any residue, and on the other hand you should be able to deduce the sequence in the right orientations. So, you can imagine that you have a biomolecule. So, all these balls what you see is the monomer which are being associated with each other. So, and in addition to that, sometimes the biomolecules are also being having the intramolecular bondings. So, these intramolecular bondings also are very important, and especially when you are talking about in terms of the proteins. So, if you would like to sequence this biomolecule, which is actually made up of these many substituents or the monomers, what you are going to do is, first you have to destroy all these bondings which are actually intramolecular binding, and which you are going to destroy. The second thing is, you also have to cleave these particular biomolecules at every amino acid or every monomer, at the end of every monomer. So, what will happen is that actually, suppose this is A, this is B, this is C, this is D, this is E, this is F. So, if you cleave after everything, what you are going to get? You are going to get a combination of all these alphabets into the solution. Now, the next step is that you are going to capture these alphabets. So, what you are going to do is, you are going to put some kind of reactive groups, so that it will react with A and form a complex. It will also react with B, it will also going to react with C, it will going to react with D, and it will going to react with E. Now, what you are going to do is you are going to isolate the individual A, B, C, D which are modified, and then by using the HPLC techniques you will be able to deduce the sequence. So, now let us take a overview of what are the things or you have to do when you would like to do the protein sequencing. So, you know that protein is a very complex molecule. It also has the peptide chains which are connecting the monomeric amino acids, and on the other hand it has the sulfide bridges which are actually forming the intramolecular bondings. And as I said in the previous slide, that the first thing is that you have to destroy the disulfide linkages. So, you have to destroy the disulfide linkages simply by the reduction, and once you destroy the disulfide linkage, it is actually going to have a linear chain of polypeptide chain. Then what you are going to do is you are going to cleave this polypeptide chain with the help of the different types of the trypsin or different types of cleavage agents. It could be the proteolytic enzymes or it could be chemical reagents. Then you are going to have the option of doing a sequencing either by the Edman degradation method or this. So, then you will do the sequencing, and that will actually going to give you the sequence of all these small fragments which you are going to generate after the cleavage. Now, all these and on the other hand, if you would use the chemical molecules that such as cyanogen bromide, that is also going to give you the different types of fragments, and all these fragments then you are going to put them together with the help of the different types of softwares, and that actually is going to give you the original sequence. So, this is what we are going to discuss today, and in following this story, the first step is that you have to destroy the disulfide linkages. So, in the stage 1, when you start with the protein, you have to break the disulfide bonds. So, disulfide bonds are always been present between the two cysteine residues, and what you are going to do, you have the two options or the two approaches. And why there is a requirement of breaking of disulfide linkages? Because the disulfide linkages always interfere in terms of the final sequencing or setting or actually deducing the sequences, because it does not allow your monomeric sequences to put together. And on the other hand, the disulfide linkages also interfere in the cleavage of these particular peptides. That is why in the first step itself you have to remove the disulfide bridges. So, you have those disulfide linkages actually interfere with the complete sequencing protocol, as it does not allow the release of the cleaved amino acid from the peptide chain. So, there are two approaches to disrupt the disulfide linkages in a protein sequence. So, you have the two approaches. In the one approach you are using the DTT, and in the other approach you are using the oxidation by the performic acid. In the first approach, the protein is oxidized with a performic acid to produce two cystic acid residues. So, in the first you are going to do oxidation with the performic acid, that is going to break the disulfide linkages, and that actually is going to give you the cystic acid. And on the other hand, in the alternate approach, what you have is that protein is reduced by the DTT, and once you or beta mercaptoethanol, both are actually the reducing agents. So, you reduce the disulfide linkages. Because of that, what will happen is that SS bond is going to be get converted into SH, and the linkage between them is going to be broken. But when you convert these by reduction, the SH is spontaneously going to come together and form the disulfide linkages. To avoid that, what you are going to do is you are going to treat this with the iodoacetamide. So, you treat this with the iodoacetamide, and that actually is going to create or generate the carboxymethyl cysteine, and once it is generating the carboxymethyl cysteine derivatives of the two cysteine present on the protein, then these two molecules will not come together and it is not going to form the disulfide bonds. So, these are the two approaches what you can use to break the disulfide linkages. In approach 1, you are using the performic acid which is going to give you the cystic acid, and in the approach 2 you are going to do the reduction with the DTT or beta mercaptoethanol, and then followed by you are going to have the reaction with the iodoacetamide. Now, let us move on to the next step. So, in the stage 2 you are going to do the cleavage of the polypeptide chain. So, once your disulfide linkages are gone, then the second step you are have to cleave the linear polypeptide chains. The cleavage can be done simply by the proteases or the chemical agents. These are the some classical proteases which people are normally using for generating the fragments such as trypsin, chymotrypsin, pepsin, or the chemical agents such as cyanogen bromide. All these molecules are having a specific cleavage reactions. For example, if you take the trypsin, it is always going to cleave just after the lysine. So, if suppose you have a protein like this, then if you treat it with the trypsin, and as I said, like trypsin is going to cleave just after the lysine, then it is going to cleave here. Okay. And if there is a arginine, then it is going to cleave just after the arginine. So, if you treat this particular peptide, what you are going to get? You are going to get two fragments. These are the two different fragments what you are going to get if it is a 7 amino acid long polypeptide chain. Similarly, the chymotrypsin, it is going to cleave after the phenylalanine, tryptophan, or the tyrosine. Similarly, the pepsin is a more non-specific proteases, but it is still be going to cleave after the leucine, phenylalanine, tryptophan, or tyrosine. Whereas, if you use the cyanogen bromide as a cleaving agent, it is going to just cleave just after the methionine. So, whatever the enzyme you will use for cleavage reactions, it is going to chop off the protease, or it is going to chop off that particular peptide or protein sequence into the individual small fragments. Now, you will move on to the stage 3. So, in the stage 3 you are going to do the sequencing of these small polypeptide chains, and so once the peptide fragments are generated, we can start the sequencing of each polypeptide chain. It has the following step. So, the first step is that you have to identify the N-terminal residue. Okay. So, every polypeptide. So, for example, if you started with the one protein sequence like this, and then you suppose you cleave this with the protease, you are going to get multiple fragments. Now, all these multiple fragments are going to be the sequence individually, and the first thing is that you have to identify the first amino acid of all these polypeptides. Okay. So, the identification of the N-terminal residue is a 3-step process. In the step 1, you are going to derivatize the terminal amino acid. The chemical reaction is performed to label the terminal amino group with the compound such as the Sanger reagent or 1-fluoro-2,4-dinitrobenzene DFNB, which is actually this. So, what you are going to do is you take the DFNB and react it with the your polypeptide chain, and the N-terminal amino group is going to react and the densyl chloride. In the most of the cases, these reagents also label free amino acid present on the basic. So, these reagents are going to label the amino group present on the basic amino acids such as lysine and arginine. So, the dinitrofluorobenzene react with the free amino groups to form the dinitrophenyl amino acid complex. So, once you do that, there will be a release of hydrogen fluoride, and then it is going to form the complex with the terminal amino group. So, that is the first step, derivatization of terminal amino acid. Then you move on to the second step. In second step, you will hydrolyze. So, you will hydrolyze this particular peptide complex, then so acid hydrolysis of the dinitrophenyl amino acid complex leads to the breaking of the peptide bond to release. So, once you do the acid hydrolysis, the peptide bond is going to be broken down between the R1 and R2, and as a result what will happen is the first amino acid is going to be released from the along with the complex. So, the dinitrophenyl amino acid complex is going to be released and the remaining peptide chain will remain intact, and then the third step is the separation and the analysis of derivatized amino acid. Then what you do is you take this whole reaction and inject it into a HPLC machine or the TLC plates, and that is actually going to separate the complex, and with the help of the amino acid or the standard amino acid, you could be able to identify this particular amino acid. So, what you are going to do is you are going to run the standard amino acids as a complex, and in addition to that you are also going to run the reactions. So, that if you compare these two chromatograms in the HPLC, or if you compare the TLC plates of these two reactions, it will tell you that which amino acid is this. In an alternate method of sequencing of the polypeptide chain, so this method what we have discussed so far, this is the method what is being developed by the Sanger, and that is why it is called as the Sanger sequencing technique. Whereas, in the alternate method what you are going to do, you are going to do the similar steps as we have done in the Sanger sequencing method, but with the different sets of reagents. In the Edman degradation sequencing method, you have the following steps. Similar to the Sanger reagent, the phenyl isothiocyanate, which is actually this, reacts with the terminal peptide chain or terminal amino groups to form a cyclic phenyl thiocarbomoyl derivatives. So, in the step 2, under the acidic condition, the terminal amino acid is cleaved from the main chain as a thiazolene derivatives, and once the thiazolene derivative is extracted into the organic solvent and it forms the phenyl thiohydrotoin amino acid PTH amino acid complex, which is this one. In the presence of acid, the PTH amino acid complex can be identified by HPLC or the TLC in comparison with the standard amino acid. Step 1 to 4 can be repeated again and again. So, you can imagine that you have a peptide chain, the terminal peptide is going to react with the phenyl isothiocyanate, and it is actually then going to label the terminal amino acid to form a cyclic phenyl thiocarbomoyl derivatives, and the peptide chain is going to be cleaved from the main chain, and this particular derivative, if you extract it into the organic solvent, then it will actually going to form the PTH amino acid complex, and this is for terminal amino acids or terminal residues. Now, what you have is you have the PTH amino acid complexes of this particular complex. So, what you can do is you can take this and run it into the HPLC, and or you can load it onto you can spot it onto the TLC plates, and along with that you can also run the standard PTH amino acid complexes, and by comparing these two chromatograms or the TLC plates, you could be able to identify the terminal amino acid, and then what you can do is you can repeat the 1 to 4 with the remaining peptide chain, and that is how if you continue in this particular cycle, it will tell you the full sequence of that particular peptide chain. Apart from that, so far what we have discussed about the sequencing of the N terminal amino acids. You can also do the C terminal sequencing, because the protein has the two groups, is the amino group as well as the carboxyl group. So, there is no method available for sequencing the carboxyl residues or the C terminal residues, except that you can actually get the C-terminal amino acid residues simply by if you treat the protein with the carboxypeptidase. So, if you know that the carboxypeptidase is a specific protease which actually chews up the protein from the carboxyl side, which means it is chewing up from the reverse side. So, in those cases what it is going to do is, so you have the protein which is actually having the N terminus on one side and the C terminus on the other side. So, under ideal condition, when we are treating this particular protein, we are treating it with the normal protease, and normal proteases are cleaving the protein from this side. But if you are treating this with the carboxypeptidase, the carboxypeptidase is going to start cleaving it from the C terminal side. So, because of that, it is going to release the C terminal amino acid, and then once this particular amino acid is going to be released, then you have the option, either you use the Sanger's method or you can use the other method to identify this particular amino acid, and that is the only way you can be able to get the identity of the C terminal amino acids. Now, in the stage 4, you have to do the ordering of the peptide fragments. So far what we have discussed that you take the protease, you take the peptide chain, you first remove the disulfide linkages. Now, it is going to be a linear peptide chain. Now, what you do is you chop off this particular peptide chain with the different proteases. Now, you are going to have the small peptide fragments. Now, these peptide fragments you have sequenced either by the Sanger method or the other method, and then now you have the sequences, but these sequences are not going to give you the original sequence from where you have the protein sequence. Now, the second step is you are going to put these peptide fragments into a particular order, so that you will be able to get the original sequence. The usage of different protein cleavage reagent produces overlapping amino acid stretches, and these stretches can be used to put the whole sequence. Let us understand this. Now, suppose you have taken a protein, okay, you cleaved up into this, like so, it is going to have the 4 fragments: fragment number 1, fragment number 2, fragment number 3, fragment number 4. Now, let us see how you are going to get the fragment. This is the fragment number 1, this is the fragment number 2, this is the fragment number 3, and then you are going to have the fragment number 4. But when, so you can imagine that a protease is cleaving it at 4 sites, but what you are going to have is also this, which is actually the fragment number 1, 2. You are also going to have a fragment which is this, which is 2 and 3. You are also going to have a fragment which is 3 and 4, and so on. So, you can see that you are not going to get the 4 fragments, but you are going to get first fragment, second fragment, third fragment, fourth fragment. You are also going to get 1 and 2, you are going to get 2 and 3, you are going to get 3 and 4, and in some cases you might get 2, 3 and 4 as well. But 2, 3, 4 is going to be slightly bigger, so that may not be very helpful, because you are not going to use those particular larger fragments to sequence. But you can see that if I have 1 and 2 separately, and I also have 1 and 2 together, I will not going to take much time to know that whatever the amino acid is present on the end of the first peptide fragment and the beginning of the second fragment is going to be the same amino acid, and in those cases it will be very easy for me to put these 2 fragments together. Similarly, if I have 3 and 4 separately and I have a overlapping stretch which is covering the 3 and 4, then it will not going to take much time to understand that this is the junction point, and that is how you are going to put all the 1, 2, 3, 4, and that is how it is going to be placed one after other, and that is how you are going to have the full sequence ready. The stage 5, you also, since we have broken down the disulfide linkages, okay, so you also, since protein sequencing means you are going to generate the original protein sequence. So, you also should have to know that where the disulfide bonds are present. The protein cleavage by the trypsin is performed with or without breaking the disulfide linkages. So, to locating the disulfide bonds, what you are going to do is you keep the protein with trypsin in the without or with breaking the disulfide linkages. Now, when you do the protein sequencing analysis, the fragment will provide the site of the fragments. The presence of one disulfide link will reduce two peptide fragment and will appear as one large fragment. You can understand this if you have a peptide sequence and you are having a disulfide linkages, and suppose I am not going to break this disulfide linkages, then what will happen is, if I am cutting this from here, I am going to have this large fragment, whereas if I do not have this, I am going to have the two fragments. So, if I do not do the removal of disulfide linkages, and even suppose I cut the with the trypsin, then I am going to get a large fragment, and that kind of analysis is going to tell me the position of the disulfide linkages within that particular protein sequence. Lastly, because we have to deduce the sequences, there is a method which is people are very often using is mass spectrometry. So, in the recent past, mass spectrometry in conjugation with the proteomics information is also been popular tool to characterize the each peptide fragment to deduce its amino acid sequence. And you can, if you are more interested to know this technique, you can be able to read this particular research article, and that will actually tell you the potential of mass spectrometry in conjugation with the proteomics information. And there are a lot of exciting courseworks or course material is also available about the proteomics in NPTEL as well as the other online web sources, and that also you can be able to consult if you are interested to know about the potential of mass spectrometry in deducing the peptide sequence. So, with this we would like to conclude our lecture here, and in our subsequent lecture we are going to discuss about how to characterize the functional groups with the help of UV visible spectroscopy as well as the IR spectroscopy. Thank you.